Hello and welcome to Bangalore International Centre's very own podcast, BIC Talks. Bangalore International Centre is a platform for informed conversations, exchange of ideas, and a space for arts and culture. BIC Talks brings the essence of all that the physical space stands for and more to your doorstep.
Hi, I'm Pavan Srinath and welcome to BIC Talks. While COVID-19, the SARS-CoV-2 virus and their impact on society dominate our lives, our fears and the airwaves, it's also important to take a step back and understand what viruses are, how they've evolved, and how they play so many roles in our lives and on Earth, beyond causing infectious diseases that also can kill humans and animals. Pranay Lal joins us today to talk about viruses. Pranay is a biochemist who works in public health and environment. He is also an author. Apart from numerous publications and articles, Pranay has also written Indica, a deep natural history of the Indian subcontinent, a marvelous book that should be enjoyed in its hardcover. We'll have Pranay talk about Indica later, but today Pranay is here to talk to us about viruses. Pranay, welcome to BIC Talks.
Thank you for having me.
Pranay, why do you love viruses when we are seeing these viruses that are causing all kinds of havoc in our society?
Pavan, to me, there is an undeniable beauty in the way viruses go about their lives. Outside of a host's body, a virus is inert and it only springs back to life when it finds a host in which it can inject a tiny package of genes. And to do this, it has to overcome a maze of complex defense systems of a host. And what a virus has to do is not only find a specific host, but also find a very specific cell. And even on the specific cell, it has to find a very particular protein on which it has to latch on to. So it's a bit like a alien spaceship, landing on a cell. And, it's like finding the right place where it can dock itself. And its own protein system has a key and it will latch on to the receptor cell of the host. In this case, let's imagine it to be a human lung cell since we are in the SARS-CoV-2 business. So it uses that lock and key arrangement and opens that latch and injects its genetic material. And that's where the whole rhapsody of the virus begins. Now, viruses can be either DNA or RNA, and there's immense complexity. They can be double-stranded DNA or single-stranded DNA, and same thing with double-stranded RNA or single-stranded RNA viruses. There is also, the length and the size of the virus. They can be of immense diversity. And what is amazing is that we just don't know so much as we should be knowing about viruses. My first idea of having seen a virus was very dramatic. I was a master's student and we had the opportunity to go to the Tata Institute of Fundamental Research in Bombay. And we saw electron microscope for the first time. And we were seeing a virus attack a bacterial cell. This bacterial attack by a virus is done by a very specific virus called a bacteriophage. When you see it for the first time and you don't see it moving because it's a snapshot, but you see several bacterial cells being in different phases of attack. And actually, you just have to let your mind go a little free and try imagining what is happening, because there is a virus which is already in the process of injecting the DNA and the others about to land. And, there are different phases. And so for a moment, if you were to just let your imagination let loose, you could actually get a sense of the rhapsodic cycle by which a virus attacks the bacterial cell. And, for that moment, I had Strauss in the back of my mind and, the brass band of Reich in the back of my mind. And Sibelius drums were rolling, doom, doom, doom. It was absolutely captivating looking at how viruses invade a cell. So I think
this is like the ride of the Valkyries.
Absolutely.
Playing in the background while viruses are invading bacteria.
Absolutely. I mean, no doubt about it. It's drama in real life. The only thing is, I wish, there are now very good electron microscopes that actually do live recordings. So unfortunately, there are very, very few of them in India, but it is great fun to actually sit and watch the whole process. I might be sounding a little pathological and morbid here, but the fact is that we know very, very little about the virus itself or even the nature of viruses that exist. And each year you find several new varieties of viruses. So my appreciation for viruses as a whole is that we as humans need to learn and understand the power of viruses. I think we need to understand them better so that we can anticipate not just outbreaks, but what else do they do in terms of managing and manipulating the ecosystems. I think only if we understand those processes do we, in some way, begin to understand what an outbreak or an epidemic or a pandemic would look like.
Right. So it's almost like saying that because mosquitoes are one of the most, are vectors and therefore result in more human deaths than perhaps any other animal out there, that doesn't mean that we should start hating on all insects rather than studying them, exploring them, and loving them in all their beauty and all their glory, right? I mean,
Oh, absolutely. Well, I think what we are seeing that assault, and yesterday was World Biodiversity Day and also World Bee Day. And bees are the biggest casualty of our hatred for insects. I mean, we use indiscriminately. We fog every evening. I don't know if that's the case in Bangalore, but surely in North India, in Delhi, in high society, and even in slums and shanty towns, you've got these fogging machines going all across the area, dispersing noxious chemicals, many of which are banned. We know that they're not useful for killing the mosquito. It makes very little impact on the mosquito's population. It is only done for mitigating public anger against the rise of or the growth of mosquitoes. So I think, yeah, your analogy of hatred for mosquitoes, which are vectors. But I think in the case of SARS and also now SARS-CoV-2, bats are being incriminated. And I think it is tragic because bats themselves, I mean, most of our tropical fruits, example, figs and certain species of mangoes and many, many other varieties of tropical fruit would not exist if we did not have bats or bees for that matter. And even mosquitoes, let's be very, very clear about it. Only 7% of all mosquitoes that we know that exist feast on human blood. So it's really not that we have to go out with a can of pesticides and start killing them. So I think that's where we are getting our story wrong. But my takeaway is this, that for us, more importantly, if we truly love nature and want to understand the complex way in which it works, whether it's a virus or the processes with which a mosquito or a bat operate, we must begin to appreciate the diversity and inventiveness of each of these creatures, especially, say, viruses, because that's one of the most understudied and least understood creatures that we know. And the same thing is also with, of course, insects and fungi and, all the small things that we just don't understand and therefore don't like. And the reason why we need to do that, Pavan, is because if we understand them better, we can become witnesses and not victims to the lethal force of nature that we are facing now. And my understanding is that there has to be greater investment and greater interest, right from not just the handful of scientists who say study bats or mosquitoes, but also the way the science is communicated to the public and therefore policymakers. And I think that is more important. Yeah.
so, Pranay, I wanted to ask you, we've had a lot of research and a lot of speculation, a lot of work that has gone into the origin of life, right? I mean, how did life first evolve? How did we get those first few molecules that could get together, perhaps self-replicate, right? And that also dives into the definition of what life is. Now, we don't have a clear picture yet, but where do you think viruses came into the picture here? Because viruses do not live on their own, cannot survive on their own. They need a host and the host machinery to function. So they're intrinsically parasites. So, like we ask the question, did the chicken come first or the egg come first? Did the virus parasite come first or did the host come first?
Pawan, I think you're right that we may never know how life began and the early building blocks and what was that process, because I think truly we'll need to go back in time to do that, and that, as you know, currently is not a possibility. But what we know through understanding from molecular studies, I think what we've been able to piece together is that there is a lot of shared genetic material between different creatures. And I think that's which is the point that actually points us to the fact that there is a shared common element that is within the tree of life, which links disparate creatures with each other. Now, what we know is that viruses perhaps emerged around the same time as the first cells and became the drivers of evolution by transferring genes from one cell to another and therefore spurring evolution. So they became drivers of evolution by constantly juggling genes between them and across new cells. That's one way to look at it. And there are astrobiologists and exobiologists who say that they could have also come from on meteorites and other extraterrestrial bodies that could have carried them. That is a strong possibility because we do find very basic amino acids on meteorites, but that really does not mean that they formed there or did not form there. So that jury is still out, and I think it's going to be a matter of debate for another 100 years or so, and there may still not be a definitive answer.
Pranay, I want to pause you there once because you mentioned how viruses can take genes and bits of DNA and RNA from one organism to the other. Can you tell us a little more about that?
Let's put this in perspective. So, if you were to look at what has been the driving forces of evolution in the world, perhaps sun and plate tectonics come right at top because, the way the sun radiates over millions of years or even hundreds of years or perhaps even single years can shape the evolutionary path of certain creatures or decimate some and support the others to grow or foster better. Plate tectonics, because the way the land arranges itself around water or the proximity of one land mass to another, and whether it's a hill or a deep valley or a gorge, all those things also determine the fate of
The third would
species.
probably be the atmosphere, right? And
Also,
whether it had oxygen or something else.
yeah, absolutely. When I say sun, I think it takes the whole package.
Fair
It's
enough.
got atmosphere. There has to be gravity. I mean, you could have atmosphere, but it dissipates if there's no gravity. So I think those Goldilocks conditions are kind of considered. And I mean, having considered those, I think plate tectonics, because you could have water and everything, but if you have an immobile Earth surface, chances of you evolving continuously may be diminished. So I think for me, the power of the sun, solar energy, atmosphere, all those together, then plate tectonics, and then the third real force of what spurs evolution are viruses. Because viruses do one thing, like I was saying earlier, is that we share genes with several creatures. There was this, in 2003 we made a remarkable discovery with the Human Genome Project. It was the first time that the human genome was sequenced and was completely read by scientists, by geneticists. And what was discovered is that near complete sections of genetic material from RNA viruses were found embedded in our DNA. Now, so this was kind of mind-blowing because what we now know is that within our genetic material, we've got long sequences of RNA embedded in our DNA from a family of viruses called the retroviruses. Now, retroviruses are very common because we know retroviruses, one of the leading members of the retrovirus is, of course, the HIV virus. And what this virus does is it converts its RNA and embeds itself into the DNA of the host. And as it does this, it continues to support the host's genome during the replication of that process. So what happens is in subsequent generations, the human genome from whichever generation it was would continue to take that embedded RNA into the human DNA and take it down generations. So one of the creatures that they discovered, the virus that they identified, was a disease called the Borna virus disease. And
This is BORNA,
yeah,
Borna.
it's Borna disease virus. Now, Borna is a place in Germany. Yeah, and the disease was first discovered in horses. And we found a near intact Borna disease virus RNA within the human genome. But as geneticists and other molecular scientists continued to study other animal genes, they found that the Borna gene was also there in primates and in certain bats and was found in elephants and fish and lemurs and rodents. So the question is, how did it get there? And so one thing that scientists imagine is that it's slightly unproven hypothesis, but it is a hypothesis nevertheless. Perhaps the one which is most tenable is that the genes got embedded and spurred evolution of each of these animals onwards because retroviruses were able to convert the RNA genome into DNA and stay embedded within that genome. So that is, I think, fascinating. And I think this is something that we've only just begun to understand in 2003. And, the real impetus of the Human Genome Project was to identify disease and disease burden caused by genetic markers or mutations within the human genome. So the focus has largely been there. And I think it would be very interesting for us in terms of understanding the evolutionary force and by studying the impact that has had historically on the human genome. So we need to now study more intensively what has happened in terms of in the past.
So basically, this means that usually when we think of evolution, we think of mutation that is being passed on from generation to generation, especially in organisms that simply divide, right? Like a bacteria becomes two bacteria. And we have heard of some things like gene transfer, where, one bacteria can share genes with another, etc. But by and large, it was this and sexual selection and mixing, right? So we inherit half our genes from our mothers and fathers, and therefore that defines our genome. But this becomes a completely new sort of X factor that comes in. We don't know when, we don't know where, but it can come in at various points in our history and perhaps dramatically change the course of a species and its evolution, right?
Well, truly, I think, one thing that the Human Genome Project estimated was at least 8% of human genome is derived from retroviruses. So if that's huge, and another 2% was attributed, that was in 2012 and 2013, to our immediate ancestor, the Denisovan ancestors of humans. So, basically, we are not, who we are. We are not so special after all. We are just an amalgamation. We are a motley of DNA pieces or RNA pieces that have been just stitched together and made us who we are. So there's another very interesting story about there's a virus called the foam virus. It actually causes a foamy mouth disease in cattle and certain primates. And they found that one of the first creatures to actually develop a very strong forelimb was the ancestor of the coelacanth, right? The deep sea living fish, which has been around for about 350 million years and was discovered off the coast of South Africa and Madagascar. And then they found it in Indonesia.
One of our living fossils that are still...
Yeah, and the fact is that it is an amazing creature, lives in depths and has a very mysterious lifestyle. But, the foam virus RNA was embedded in the DNA of the coelacanth and certain mammals, primates. A small trace has been found in humans as well. So, here's an example that we have either had this transmission in several generations. Has it been handed over to us from the coelacanth to the primate and then to humans? We don't know that process, and I think we need to understand that. We know that certain human diseases are also seen in different creatures, including fish, and there is no intermediaries in between, right? So I think what we need to figure out as we progress with technology and science is that where in the evolutionary space do viruses fit in, and do we accord them enough importance in terms of the work we want to do in the future when it comes to public health and preventing the next pandemic? So I think that is a very crucial thing for us to understand.
Thanks, Pranay. And that is still a utilitarian view, right? That if we study this, it'll help us. And there is an intrinsic value to understanding our biodiversity as well, where we may not be able to tie it to an immediate utilitarian purpose right now, but it might have one in the future, or it might just expand our minds and our thinking. And if I'm not mistaken, it's sort of similar research that led us to the technology of CRISPR as well, right? I mean, when we're talking about,
that's right,
now us being able to go and manipulate DNA, human DNA,
right,
we are essentially talking about viral technology. It's technology that viruses had that we now have enough tools to adapt and perhaps use to our own ends. And now we are trying to use some of those same tools against the SARS-CoV-2 virus with some of the vaccines that are being developed, with DNA vaccines, RNA vaccines, and more. Right?
So yeah, I think one thing. I mean, you bring a very interesting point. So as scientists begin to discover kinds of viruses, they're also finding new ways to make new tools, not just to control viruses themselves, but also control bacteria, but also to deliver genes. And in some, there's a very fascinating project in between Europe and certain other countries. The CERN laboratories are working on it. Is on nanomaterials using understanding the structures of viruses. But let's come back to something very interesting that you said, that we need to, how do we design and how do we look at improving designs of vaccines and things that will protect us. But so one of the enigmas of entomologists had was that they could not figure out that how is it that caterpillars, that we see mass roosting of caterpillars in certain seasons, and we find that parasitoid wasps go and inject them with an egg, and the larva of the parasitoid wasp feeds on the caterpillar. Now, that's a very fascinating thing, but what was not easy to understand is that caterpillars have a fantastic immune system. They have a very evolved immune system. And how is it that an egg, which is an external, extraneous, alien thing, that has got embedded in the folds of their skin, or the outer layer, would then emerge and evade the immune system of a caterpillar? Now, that is something that baffled a scientist for a very long time. And then they discovered what it was. They found that there is an RNA virus that helps this egg and the hatchling of a larva to evade the caterpillar's immune response. And it's from understanding from those enzymes that CRISPR was born. The whole story of CRISPR and gene silencing begins from our understanding of how certain insects are able to evade the immune system of another insect on whom they lay their egg and parasitize. So I think the whole understanding of the ecology of the virus, I mean, the fact that we found the virus was an accidental discovery, and therefore we got to understand the whole process of how parasitoid wasps end up infecting caterpillar or any other host for that matter.
Amazing. And now it has become the bedrock of a whole host of technologies, not just for use in research and in labs to answer other questions, but potentially for therapeutic uses as well. And people are working really hard on making those therapeutic uses a reality. So here, Pranay, I also want to ask, so like you mentioned, so viruses now form a part of our very DNA. We also know today that human beings are not just a single multicellular organism, which is a higher mammal, but we are also a biome unto ourselves, right? And we talk quite a bit about the microbiome. Now we all know that our gut bacteria are very important for our gut health. But here, so can you tell us a little bit about the human viruses that sort of dwell within us? We only hear about these viruses that come in and infect us and cause a lot of trouble. But there must be many which don't, right? And which coexist very happily with us.
Right. So, Pavan, this is when the word virome was coined, it was, it was an imaginary thing because it was like an X factor when you could not explain something in, say, cancer or, many other of the other things. It was like X-ray. It was the X factor, right? So therefore, there was very little research on the virome, trying to understand the correlation between the virus and the bacteria and the chlamydia and the fungus and the gut cells, or the host's own ecosystem of the organs.
And in biology, we usually make something an ohm when we refer to like a collection of them, right?
Collection, yeah, organisms that work together, or and there was another word that came out which was called virosphere, which is in a larger context connecting different viromes. And so what has been interesting is that we now are beginning to appreciate that the virosphere, or that connects different viromes, may be interconnected. and the fact that there are several diets, different types of diets, we have a different microbiome, right? And there is very interesting research that is being done by both private and public sector companies and organizations, which is showing how different set of parents create a child, and the child may have a distinct microbiome. That Yeah, so there is placental microbiome which is distinct, and and then it comes, when the child grows, it becomes uh, something which is intermediate to the mother and the father, or maybe completely distinct. So that's something that is coming out quite differently, and I think our understanding there is only growing. But what is crucial is, again, the focus is on largely on bacteria, because the focus for at least for private sector is to look at how they want to look at foods, for example, or nutraceuticals, to help improve diet and retention of micronutrients. So while that remains the focus, the larger thing that we are missing out is, again, the virus. Because for my understanding is that, think about it in this way, that we've got, for example, E. coli in our gut. Now, E. coli is Escherichia coli. It's the most abundant organism in the human landscape, if we can use that word in some sense. I mean, it's there in our sewage because it's there in our fecal matter. It's there in our gut. It is an essential organism because it helps us digest several kinds of food, and very importantly, it is a major source for vitamin B, vitamin B12 in particular, in our gut. What we know is that they reproduce rapidly within the human gut. So what is it that controls, what brings in the equilibrium for organisms like E. coli? And they've only recently discovered a series of new viruses that actually bring in that equilibrium. So as soon as the population begins to increase beyond a point, they start culling out the ones that are excessive. Um, Otherwise, what would happen is that we would possibly bloat so much, because our gut is, there's a lot of activity. There's decomposition, there's fermentation, there's absorption, there's recycling of nutrients, and there's a immense amount of activity that takes place there. And anything that goes wrong with the microbes there could cause our stomach to bloat, and possibly even bloat us to death. I mean, you never know. But so there is a correcting factor, and it's certainly not our cells alone. There's many other things there that are working in a sense, which are creating that check and balance for organisms not to grow out of proportion. So I think that is an area which we now have to start beginning to understand, and it's going to be a very crucial area for us in the future.
One of my takeaways from what you've been telling us is that we think of humans as colonists and colonizers at our worst, but viruses seem to be prolific and cosmopolitan in how they can jump from species to species, influence their behavior and their evolution, and more.
Yeah, I think, Pavan, you got it spot on. I think, if you were to imagine, is it that we are the masters of the universe, or are we being played by the virus itself? Because, for every creature, there is a virus, right? And, like I mentioned earlier, their complexity, and as new ones are being discovered, we just find that there's such immense diversity that we find in not just in their shapes and sizes, but also in the mechanism in which they evade immune systems and infect. But despite this, we've only discovered about 5,600 viruses so far. And considering that this is less than the known existing mammals in the world today, now we know that as humans, for example, there are about 180 viruses that affect us, right? And if you were to lump them together, a significant number is in pigs and poultry and bats. So you might have one or two for the major other species. There would be so many others for which we will not have had discovered a single virus. Now, what I find completely insane about viruses is that, not only do they drive the evolutionary process in, very, very incremental basis on a perhaps every hour, every minute, but certainly on a daily basis, because they are creating mutations or point mutations in several creatures around every ecosystem. Now, you had mentioned chicken and egg earlier, and in evolutionary terms, it's neither chicken nor egg, because the egg can be infected also, and the mother, while laying the egg, could be infected, or the father, while giving his sperm, would also be affected. So, there could be transmission errors, those kind of things that create new opportunities for the virus to manipulate the egg. So, the chicken or egg is not a condition. It is chicken and egg, because intergenerational differences that happen during evolution is something which is not very linear. It goes into various other modes. And I want to bring this story in about the discovery of tulips. Now, we knew tulips, for example, existed in Turkey for several millennia. And it was just another plant. It was a bulbous plant like a lily that grew along agricultural fields. But it was
I'm learning even this for the first time because I thought tulips were from Holland.
Well, no, actually, if you were to look at the origins of plants, they emerged somewhere around the Levant and concentrated around the Turkey area, Anatolia. So it was taken to Netherlands, by the Dutch. It reached through Venice. It reached to Holland, and something remarkable happened in Holland. And this is, around 1550. There was a growing market to the tulips that were growing in Holland. And that's the time when, if you remember, the banks were also emerging and the shipping industry was growing hugely. And the first market that emerged in Amsterdam, around Amsterdam, was in 1604. I don't know if you remember, but in medieval history, Holland and that part of the world was called the Low Country because they were much below the sea level, and there are dikes that prevent the ingress of water from the sea. So what happened was that, because this was being planted into a new place, a place which was very different from the localities where it was planted earlier, there was a virus that actually caused streaking and banding of tulips that were growing here. So in 1634, they found a beautiful variegated tulip variety which became a craze. And it started from one farm just outside of Amsterdam. And it was so pretty. It was dark blue in color and had this yellow golden streak on it. And later they found that the number of the bulbs that were producing the same quality of flower was declining. So in 1637, there was this, very small patch of about 80 bulbs or 80 flowers, if you want to say, that grew of this flower. And there was such a rage by that time that there was a ship that was actually given, a seaworthy ship was exchanged for those 80 bulbs. Now, what that means is it's like actually giving your license to take that ship to the Indies and get pepper and more expensive things and silks and aromatic herbs and whatever. That was the price that was given to these 80 tulip bulbs. What is very remarkable is that this is the first recorded instance of economic bubble, because people started to speculate that the next crop of the red ones or the blue striated and variegated ones, and we are going to pay this price. It could be worth a bushel of wheat or 5 cows, and so there was a demand in rural and semi-urban Holland where the prospectors would actually go and bid for the commodity in advance. But this whole hype fell flat because in certain years, 1636 to 1638, the numbers of variegated tulips collapsed. And as the collapse was happening, the price of the variegated and the striated tulips grew. And it was at this time that the first bubble actually happened very quickly, and the economic bubble where you pay it forward. You forecast a price and you pay somebody in advance and a significant amount of money, right? And that's how the whole story of viruses being able to create something of enormous beauty, but also causing collapse of an economy in a sense. So that's how powerful they were.
Amazing. So it was these viruses which were even causing the striations and the eventual demise of the tulips as well?
Well, they did not cause the demise of the tulip. They just did not infect the tulip that season. So the tulip was plain. It just did not get the striations. So it was supposed to be dark blue with yellowish golden streaks, and it just stayed dark blue. It was a beautiful blue, but that yellow streak is the one that actually made it so vivid. And there was also a red, gold, and white, which was also very famous, another very popular, very expensive tulip. So it is the whole idea of striation with the virus that actually created that buzz, and therefore the speculation that people have to own one. If you are somebody in Holland, if you are a prosperous burger, as they were called, the word burger actually comes from Holland. If you were a burger, you would have at least a set of tulips in your house. It was something to show off.
Wow. So it was the virus that gave the tulip its value, its special value,
Absolutely.
enough to drive a bubble. Amazing.
Absolutely.
So, and this is centuries before we could even discover the virus as humans, right?
Absolutely. This is 1637.
And, there's one thing that strikes to me about early discovery of the virus and before even what virus was known. The word virus itself comes from the Latin virus, which is the same cognate, the same root word as wish in Indian languages, which is poison. And this was because the viruses were so small that scientists could not isolate them the way they could isolate bacteria. And so they could not determine what caused the disease. And they thought that there was some liquid poison which caused the disease, right? So even this discovery of viruses seems fascinating.
I think, this is discussion which is, the early discovery and, the fact that we knew that there was an invisible agent causing mayhem and death is a topic by itself. And I think we could talk about it another time. But, for me, the fact that people knew that there was something miasmic or something, in many cases, it was considered black magic. The fact remains that until Leeuwenhoek actually saw and Hooke saw the first organisms in soil or a drop of pond water, there was no imagination that there could be biological entities that could be that small. And viruses are extremely, extremely tiny. 1 billion of them could actually sit on a single grain of salt. And this is what makes viruses a true exception to the living world. I mean, there are clearly no thumb rules that govern any genus of a virus. And the taxonomy of viruses is even more complex than, say, beetles or fungi for that reason. For example, in 2002, they discovered the biggest virus embedded in a protozoa. It was within amoeba, actually. It's about 1,000 times larger than a smallpox virus. It is so large. So if when you stain the protozoa and watch it under, or don't even have to stain it, if you have a good, simple microscope, you can actually see it as a dark blue blob in the center of the protozoa of this amoeba, and it's called a mimi virus. It's called mimi because it's microbe mimicking, mimi virus, right? So and that's how we discovered it. And the other thing that I think which is very interesting is that a lot of people question that there are so many viruses around us, then why is it that we don't report so many viruses from India? I mean, all of those stories come from Southeast Asia or Africa or sometimes even Latin America. The sad thing is that India actually has a variety of viruses, and I don't think we should be ashamed of them because, I think public health and institutions and government like to put a cover on things, on new discoveries. And I think we have our own set of very, very interesting viruses, which can, if we invest in them, we can find new ideas and solutions and understand the viruses better. I mean, we've got the famous Chandipura virus, which is as lethal as the Ebola virus. But the thing is, we put a cover to everything. We like to sweep it under the carpet, right? And then there's the Ganjam virus. There's the Kyasanur forest virus, which is a very famous one in Karnataka, which is a tick-borne fever. So for us, and there's one of my favorites is a virus called a Thotapalem virus, which is a Hanta virus discovered in the 60s from near Vellore. Thotapalem is now a suburb of Vellore, but it used to be a village in the 60s. And, while we know that hantaviruses occur in rodents, this particular virus is found in shrews. And it's only found here in Thotapalem. You go another 60, 80 kilometers northwest or east of Thotapalem, you don't find it in shrews.
This is shrew the mammal, right?
The shrew mammals, yes. So the insectivorous creatures, that amazing creatures by themselves. So I think what is very interesting is that we should invest in, as a country, we should invest more on research on viruses. And I think let's get out of that phase where we start covering up and let's be more transparent. Let's not be cagey about looking at virus and viruses research. So I think that's something which is very important.
Certainly, Pranay. If I'm not mistaken, there are some studies which show that over half, or at least half, the fever causing diseases that are there in India are by unknown viruses where we don't yet know the source. So, more information is absolutely needed from a health perspective. I want to switch gears a bit and go back to this idea of viruses as these regulators and gatekeepers in the overall ecosystems, including the human biome. Do viruses play a similar role within our larger ecosystem and our climate?
I think we need to again understand not just the virus, but the things that the virus regulates. Now, I think we need to step back a bit and understand something very, very interesting. So, in the early 1990s, we discovered a creature. We, when I say we, it's the larger scientific community on whose behalf I speak. I mean, there was a discovery made by scientists at MIT, and in particular, a very, very important microbiologist by the name of Penny Chisholm. She discovered a creature which is a photosynthetic bacteria in seawater. It's called Prochlorococcus, and it has a cousin called Synechococcus, which also lives alongside. Now, until then, it was imagined that trees were the major drivers of carbon fixing and carbon assimilation, and also producing all the oxygen that we breathe. But the discovery of this creature, which is predominantly in the first 8 meters of warm tropical oceans, and as you move along to the temperate oceans and seas, it is in lesser numbers, but it's still significant. What happens is that this creature,
Pranay, are these phytoplankton?
they are cyanobacteria. They are like closer to blue-green algae in some sense in terms of their photosynthetic structure. So they're very, very simple creatures. They're very, very, very tiny, and possibly escaped attention of just about every scientist who was working on marine microbiology until the 1990s. But when this was discovered, it was very big news. What it proved was that when they started estimating that the population and the size of how much Prochlorococcus and Synechococcus exists in the oceans, they estimated that it possibly buries about 40% of carbon dioxide and also adds the same amount of oxygen every day. It's a humble microbe in the oceans, far away from the maddening world, and it's doing its business. And on a daily basis, it produces this amount of oxygen, captures the carbon dioxide, grows, replicates, and the population of this organism also needs to be controlled. And this is where viruses come in. Because if it continued to replicate without any controls, you would have algal blooms, the kind that you see, or the red algal blooms, or the one green algal bloom that you're seeing now in Antarctica, right? So those are the kind of things that you would be covering the entire oceans. It's a virus that makes sure that the sinking of the cell, once the cells begin to age, they will attack an aged cell. It has finished its replication duties, its reproductive duties, and it is now infected by a virus. It gets punctured and it goes down, settles down at the bottom of the sea, and that's where it gets buried as carbon burial in deep waters. Now, that is very critical. So the whole cycle of absorption and the death of the single-celled organism is manipulated by a virus, and which I think is very, very crucial, because if we are looking at the larger realm of carbon-oxygen cycle, the virus has important role to play. And I think neither the bacteria or the virus can be ignored.
Wow. And in this, when humans talk about geoengineering to tackle climate change, it feels like such hubris when we are discovering organisms like these which are regulating critical processes that maintain life on Earth as it is. And we are discovering new things every other year.
And this is the most abundant creature in the world. That's the sad part, that we discovered it only in 1990. And if you were to put all these creatures on one side, I mean, their contribution to maintaining the balance on Earth's atmosphere or the processes of the Earth is immense. What is also critical is that we now need to understand that these organisms are under tremendous stress and threat, because you've got massive ocean liners that are passing through. You've got marine pollution that is happening, not just from urban centers that are located around the fringes of the oceans, but also the amount of waste that we are discarding, whether it's through marine traffic or also deliberate dumping of waste, whether it's nuclear or industrial chemical kind of complexes. So I think we now need to seriously consider what is it that we really want to do. And I think the time for introspection is now, because if the health of the planet has to be seen, it has to be seen in its totality. And something even as simple as understanding carbon budgets and oxygen budgets, I think we have got that, the ledger of how much we need to breathe in and how much we need to exhale is also wrong, because we have never taken these creatures into account.
Wow. Pranay, thank you for taking us through the sturdy force of viruses and the various roles they play in our lives. And if I may, we talk about, ancient models of the world and how we sit upon a turtle and then what's below the turtle, it's turtles all the way down. My takeaway from our conversation today is that it's microbes and viruses all the way down.
Yes, absolutely.
So Pranay, thank you so much for joining us on BIC Talks.
Thank you for having me.
Thank you for listening in till the end. Please share this episode with a friend on social media, WhatsApp, or anywhere else. It would mean the world to us. And in case you're listening via iTunes or Apple Podcasts, please leave us a rating and a review. Subscribe to BIC Talks on email or your favorite podcast app, and don't miss out on future episodes. This episode of BIC Talks has Gaurav Krishna as our sound engineer, with support from S. Sarvanaraj and Lekha Naidu. And the accompanying episode artwork was made by Chandni Venkataraman.
Thank you for listening to this episode of BIC Talks. This podcast can be accessed on our website, bangaloreinternationalcentre.org, as well as on any of your favorite podcast platforms. Tune in for new episodes every week, and do subscribe to our YouTube channel and follow our Facebook, Twitter, and Instagram pages to stay informed on our latest updates.